Welcome to What I Did in Lab Last Week, where I tell you guys what I did in my biochemistry lab last week. So, this past lab was about enzyme kinetics, and I don't have any pictures from it because it was honestly kind of boring. But if you don't know what enzymes do, they're a biological protein which acts as a catalyst in order to increase the rate of biological reactions. So how a normal reaction works, there's a substrate that releases a product. How enzymes work, they bond to that substrate and basically make it release that product faster. So it just makes the reaction go from here to here by decreasing the activation energy of that specific reactions. So if the activation energy is up here, the enzyme makes it go down to here, which makes it basically just easier for the substrate to release the product. So basically what we did, we had this enzyme, this substrate, and a buffer, which we put the stuff in, and we measured the baseline action of the substrate releasing the product, which was a different color, and measured that using absorbance by the spectrophotometer, like last week. Then we did a bunch of different things where we changed the concentration of the substrate, we changed the concentration of the enzyme, which would affect the rate of the reaction, so we changed those to see what would happen. We changed the substrate specificity, so there's two forms that a substrate can take, basically the L form or the D form, which is bunch of organic chemistry that I don't want to explain, but the enzyme binds best to the L form, so we switched it out to the D form in order to see how that affected the reaction rate. And then we added, for the last bit, an inhibitor, which basically inhibits, as in blocks the activity of the enzyme. So by adding an inhibitor to the enzyme, it causes problems with that, either reversibly or irreversibly, and that decreases the rate of reaction because it stops the enzyme from doing what it's supposed to, which is increasing the rate of reaction. So we did all that. We collected the data by mixing all of the stuff together in little cuvettes and putting them in the spectrophotometer and timing it for 3 minutes, taking measurements of the absorbance every 30 seconds, and collected a whole ton of data points that I get to write a lab report on later. So basically, the faster the absorbance changed in the cuvette, the faster the enzyme was working, and the less change, the slower the enzyme was working. So yeah, that's what I did in lab this past week.